Polymerase chain reaction by Cheyenne and Joshua.
Have you ever wondered how scientists are able to diagnose diseases, study genetic disorders, and even solve criminal cases, all with one simple technology? Allow me to introduce to you the polymerase chain reaction procedure, or PCR. In this lesson, we are going to grasp the basic principles of genetics, examine the key concepts of PCR and how it works, discover the importance of PCR in genetic diagnostics and research, discuss the advantages of PCR, and provide at least 2 practical uses for it, evaluate PCR's implications, and finally, examine possible developments in PCR technology. Let us begin with a brief basics on genetics. Genetics is the branch of biology focused on the study of genes, inheritance, and genetic variability in various organisms. Genetics are essential when considering how one's traits and characteristics are passed down over generations. Additionally, the study of genetics can help determine a person's potential for obtaining certain genetic disorders and conditions, which is a topic directly linked to PCR that we will explore in the following slides. So, what is PCR and how is it significant to society? PCR, or polymerase chain reaction, is a widely used laboratory procedure in which scientists are able to produce millions of copies from a given sample of DNA, thus enabling them to conduct research. In PCR, a section of the genome is chosen to be amplified using brief synthetic DNA fragments known as primers, and that segment is subsequently amplified using several rounds of DNA synthesis. Now, you may be wondering how PCR is significant. Well, scientists are able to fully analyze one's genetic information with PCR without having to sample a large amount of DNA, which is why we can see the dependence of this technology in numerous medical applications such as genetic testing, forensic science, medical diagnostics, and the study of genetic illnesses.
Exploration of polymerase chain reaction. Polymerase chain reaction is a process of making many copies of specific DNA regions into test tubes rather than organisms. A polymerase chain reaction consists of 5 core ingredients. First is the DNA template to copy from. Next is the primers. They are short stretches of DNA or RNA. Third are DNA nucleotide bases, which are adenine, thymine, uracil, and cytosine or guanine. Next is Taq polymerase. It is an enzyme which adds nucleotides to the copied DNA. And lastly, a buffer to stabilize and ensure the right conditions for the reaction. All 5 PCR components are then fit into a machine called the thermocycler. In the thermocycler, a process called thermocycling is formed, which consists of 3 steps. First is denaturation. Denaturation is the process of heating up the tube to at least 94 degrees Celsius for the solution to maintain its form so it doesn't break or release. The heat then breaks the hydrogen bonds of the original DNA sample and separates them into single-stranded DNA. The second step is annealing. Annealing is when the sample is then cooled down to 50 degrees Celsius or 60 degrees Celsius. It is cooled down to enable the DNA primers to attach to the template. Last but not least, the third step. The third step is extending. The temperature increases once more and the new strand of DNA is manufactured by the Taq polymerase enzyme. Once completed, these 3 stages are repeated 20 to 40 times, multiplying the number of DNA copies. After the completion of PCR, a method called electrophoresis is used to examine the quantity and size of DNA fragments produced. Benefits and drawbacks of PCR. Benefits of PCR include the importance of usage in medical diagnostics, application of discovering diseases and viruses in human bodies, and environmental and agricultural applications, such as detecting pathogens in environmental bodies such as soil, water, and air. Drawbacks of PCR include being sensitive to contamination. Since PCR is used for detecting small amounts of DNA, it makes it susceptible to contamination.
Let's now move on to the real-world applications of PCR, starting with medical diagnostics. In medical diagnostics, PCR is widely used to diagnose viral infections such as COVID-19, HIV, hepatitis, and even tuberculosis. During the COVID-19 pandemic, PCR was used to detect the presence of the SARS-CoV-2 virus in patient samples, helping frontline healthcare workers drastically by allowing them to rapidly identify whether or not a patient was diagnosed with COVID-19. A patient who has symptoms of a given illness visits the nearest doctor in order for them to obtain a nasal swab test or a blood sample. During the COVID-19 pandemic, patients were even able to complete a PCR test at the comfort of their home. This 5-minute self-swab process helped patients save hours of their time. After the patient completes their testing, the sample is sent to a laboratory for precise analysis. Taking a swab test or a blood sample enables the laboratory to amplify the genetic contents and identify any potential viral DNA or RNA present in a patient's sample. When the results have been found, the sample is sent back to the doctor alongside a written report which states the results and status of the viral infection. Another well-known application for PCR testing is the field of forensic science. In forensic science, PCR is frequently utilized for criminal investigations. In order to identify suspects or victims, forensic experts can amplify DNA collected at crime scenes such as blood, hair, or saliva. Experts then take these DNA samples to a specialized lab in which the samples are then largely amplified, analyzed, and further compared to that of various suspects in order to determine the sole culprit. One of the first major uses of DNA profiling and PCR testing in criminal investigation was seen in the 1987 conviction of Colin Pitchfork in the United Kingdom. Colin was identified through DNA testing in 1987 after a sample matched the DNA recovered from the crime location. After PCR, amplification, and DNA testing, Pitchfork was found guilty after his DNA matched the forensic evidence. This case is significant since it was among the first times PCR technology was used in criminal court for DNA profiling. It showed how PCR, even with tiny amounts of DNA, may be used to solve crime cases.
Ethical considerations of PCR. Ethical implications related to polymerase chain reaction are patient identification, consent, and understanding the limitations and consequences of the test. Standards of using this technology require every patient's identity, requiring information about the patient's birth date, body statistics such as blood type, weight, height, etc. Polymerase chain reaction often requires samples of blood, saliva, and tissue from patients, so their consent is essential to the testing of PCR. Consequences of using PCR to mutate genetic information on a patient can result in psychological impacts such as anxiety, depression, and misjudgments. It can also result in ethical dilemmas with people. For example, if a person were to find out predispositions of genetic material in their DNA, it could cause a stir within their family and argue if they're biologically related. Future prospects of PCR. Polymerase chain reaction will be efficient and essential in the near future as a test for human health and conditions such as respiratory, gastrointestinal, and sexually transmitted diseases, and meningitis. With more development into PCR in the future, it's likely to become of important use to human lives as it helps with syndromic testing, such as testing humans if they have influenza, COVID-19, pneumonia, respiratory syncytial virus, etc. PCR is also used for detecting diseases such as cancer in blood and urine samples. In the next decade, PCR syndrome testing is expected to become more efficient and sophisticated with the help of automation, as it will help with detecting diseases in people. In conclusion, polymerase chain reaction is a laboratory process in which scientists produce millions of copies from samples of DNA. It also requires a step process to use called the thermal cycle, where it denaturates, anneals, and extends the temperature to mutate the genetic information. Medical diagnostics in PCR are in the works of discovering harmful viruses such as COVID-19 and HIV in people. PCR is also used in crime investigations in forensic sciences, as samples from crime scenes can be used in PCR to capture the culprit. Ethical considerations of PCR include the identification of patients, consensual responses, and consequences and limitations. Lastly, potential future development of PCR will be essential to figure out the good and bad in human health. The end.